Uh, Good evening, everyone. So let's wait for 5 more minutes, then we can start the session from 6:10. Okay. Hi. We will begin the session. I will start sharing my screen. You guys can unmute yourselves and let me know if you have any doubt or if someone else is wants to join. Okay. So today again in today's session, the last tutorial session we looked at different restriction enzymes, how are we producing, sticky ends or blunt ends and then joining it using ligase. Okay. Now we are producing the recombinant DNA. Now that recombinant DNA has to be put inside a host cell. Okay. Host cell of our interest. All right. So today this lecture, uh, like today's session will cover or summarize uh, the lecture videos consisting of how exactly are we going to introduce a recombinant DNA inside a host cell, and then what are the different methods to do that, and then again we will look at some sample questions. Okay. So uh, now this is the donor cell. And for delivering DNA into a host cell, definitely there should be some level of compatibility, or the recipient cell or the host cell has to be made competent enough to receive the DNA. Okay, recombinant DNA that we are talking about. So the surface chemistry of the host cell or the recipient cell, um, the charges on the cell surface can either attract or repel the DNA molecule as a result of opposite or similar charges, right? So DNA is negatively charged due to the phosphate molecules, phosphate moieties on it. So what happens is, but the cell surface of the recipient cell might have different kinds of composition. The membrane composition or cell wall composition can vary. Presence of cell wall in bacteria, fungus, and plant causes additional physical barrier to take up the foreign DNA and for the entry of the DNA. So that's why the recipient cell or the host cell has to be made competent. It has to be treated with different kinds of salts. There's a certain protocol that is usually followed to make the recipient cells competent, by making, by treating the host cell with different kinds of salts, by exposing the host cell to different variations of temperature, sometimes cold, sometimes hot, heat shock. That kind of temperature variations also help in creating pores in the membrane of the host cell so that it can take up the DNA. Okay, so we will look into it eventually. Now, mostly use of divalent cations has been the most effective chemical treatment to bring about transformation. The process of delivering any kind of foreign DNA inside a host cell is called transformation. Okay. Now, among the different cations, divalent calcium cations have been found to be the most effective one. In either only calcium uh, salts can be used, or in using or in combination with different other salts like magnesium. Okay. Combination of divalent and like this has been studied and experimentally verified that calcium salts are the most effective one. Calcium salt treatment to the host cell is the most effective one in making the cells competent. And it has also been seen that divalent and monovalent cations, this like calcium and magnesium, calcium and manganese, calcium, rubidium, and DMSO, dimethyl sulfoxide, and different alkali metals with a prolonged incubation at 0 degrees Celsius have also been found to be effective in uh, like like taking up the DNA in by the host cells. Generally, all divalent cations enhance the transformation process, and presence of magnesium in bacterial culture media increases the transformation efficiency by 15 to 20 folds as compared to the cells grown in the absence of magnesium salts. Okay, so usually in general, like daily laboratory protocols, what we usually do is we make a mixture of calcium and like treat the cells with calcium and magnesium salts. Okay, and it leads to very nice competent cells which can easily take up the DNA. Now, taking up the DNA is a different protocol, and making the cells competent is a different protocol. Okay, so before introducing the foreign DNA to the cells have to be made competent. This is what we are reading now, how to make the cells competent, how to make the recipient cells competent. So one way is to treat it with divalent cations, mostly salts of calcium and magnesium. Okay. Now, donor cells, from where the gene of interest has to be delivered or taken, and then this has to be like put inside another recipient cell. Okay. Now, we were talking about, since the last two tutorial sessions, we looked at selective, uh, some kind of selection mechanism, right? Mostly one of them is antibiotic resistance. So if the donor cells have this resistance encoding gene, some antibiotic resistant gene here, then if that particular gene of interest is taken up by the host cell or recipient cell, then the recipient cell or acceptor cells should also have the resistant, should also show the resistance to the concerned antibiotic. Okay. Now, most of you might be aware of the of Griffith's experiment, where actually the term transforming principle came into picture. So, what very briefly what he did. So there were two strains of bacteria, pathogenic bacterium. One was rough strain and one was smooth strain. And the rough strain was non-virulent, but the smooth strain was virulent. Non-virulent ones, when the cells were injected into mice, the mouse lived happily, and when the smooth strain of the bacterium was injected inside the mouse, it died. But when the same smooth strain, virulent smooth strain, was heat killed and then injected inside mice, it lived happily. But when they put inside a mixture of rough strain, which are the non-virulent ones, but heat killed smooth strain, the, like only rough strain, non-virulent ones, mouse, it didn't affect the mouse, and heat killed smooth strain also didn't affect the mouse. However, when rough strain, the non-virulent ones, and the heat-killed smooth strain were mixed together and injected inside the mouse, then the mouse died. So, what they deduced that some kind of transforming principle was getting, transferred to the rough strain, or something was getting transferred into the mouse that is leading to this virulence and it is dying. So transforming principle, this term came into picture after this experiment. So mechanism of transformation, this is from the lecture slide only. Transformation is the process by which cell-free DNA or any kind of foreign DNA is taken up by another cell. It can be bacterium, it can be plant, it can be human cell line, any kind of cell. Okay. So what happens here is donor cell is there. So first, if this is the gene of our interest that we want to introduce into the host cell, then you have to first extract the DNA from the donor cell. Okay. So again, you have to lyse the cells and then take the DNA out. And then this is the competent recipient cell. The donor cell's DNA, the competent recipient cell takes up the donor DNA, and then this particular DNA is now um, inside the recipient cell, and this, when the cell divides, then eventually um, some of the daughter cells might have nothing of the, like, might not have the recombinant DNA, and some cells will have the recombinant DNA, and those cells having the recombinant DNA, harboring the recombinant DNA, are called transformed cells. Okay. Now, the repulsion between foreign DNA and the bacterial cell is predominantly due to the negative charges on them both. And how is this overcome? By using divalent cations. Okay. This is applicable for linear DNA fragments as well as circular DNA molecules like plasmids that we looked earlier also. We looked at plasmid extraction as well. Okay. And the different types of plasmids that can be seen on a agarose gel. Now, divalent cations bind both to the cell as well as the DNA, because if there are negative charges on both, like DNA is definitely negatively charged, but the cell that we are talking about, recipient cell, can have both negative as well as positive charges. So right now we are talking about the scenario when both the, I mean, the bacterial cell surface is also negatively charged and DNA is definitely negatively charged. So divalent cations, when we treat these cells with divalent cations, These cations bind both to the cell surface as well as the DNA, hence neutralizing the charge altogether. The calcium bound to the DNA further helps the DNA to adsorb onto the competent cell surface. Moreover, DNA binding proteins present in the cell membrane can also help in this interaction. Okay. And when the DNA is anchored onto the membrane, what happens is it is getting adsorbed onto the cell surface. Okay, recipient cell surface. So this additionally reduces the risk of the DNA getting detached from there, or the DNA getting um, uh, excluded out of there. Okay, or expelled out of there. So this kind of adsorption that is taking this interaction is actually helping the DNA to stay on the cell surface. And when further treatments, temperature variations will help in opening up the pores, and then the DNA that is already adsorbed on the cell surface, it will be easier for the recipient cell to take up that DNA. Okay. Now, further, the lower temperatures used in transformation protocols, the temperatures that is being talked about here is mostly on a daily laboratory daily laboratory work. What we usually do is we keep it on ice. We perform this addition of DNA addition, etc., on ice only, which is basically 4 degrees. Okay. So the low temperatures used in transformation protocols congeals the lipid moiety and consequently restricts the fluidity of the cell membrane, which strengthens the calcium cell surface interaction. In this way, what happens? Calcium ions bound with the cell surface as well as the foreign DNA can bring the DNA onto the cell. Okay. Now, relative association of divalent cations is more with the cell membrane as compared to its association with foreign DNA. Membranes calcium very readily, and once inside the absorb calcium very readily, and once the calcium ions get inside the cells, these are neutralized by membrane phosphates present on the cytosolic side, okay, of the cell. The binding of calcium ions to the membrane also can cause changes in the membrane permeability. And when the cells are treated with divalent or trivalent ions on ice, then it is given a heat shock. Like mostly generally it's used as 42 degrees Celsius, like directly from 4 degrees Celsius it's 42 degrees Celsius, and the time is usually 90 seconds. Okay, so for 90 seconds there's a heat shock treatment to the given to the cells after adding the DNA, which produces a temperature imbalance. Now, molecules with increased Brownian motion outside the cell are likely to push the DNA molecule inside the cell. All right. But however, it's still unclear if this kinetic force is sufficient enough to push the DNA molecules that were already adsorbed on the recipient's cell surface. Now, this particular study, this divalent trivalent treatment, temperature shock, like how exactly cells are made competent. This study was done by this group in 2008. They studied the efficacy of cooling and heating cycles by increasing the number of cycles until maximum transformation efficiency was achieved. What exactly happens is when we lower the temperature, it can contribute to protein loss, and heating when we are giving the heat shock mostly at 42 degrees Celsius, it leads to lipid loss, and thus together these cycles increase the transformation efficiency. How do these cycles contribute? Um, it contributes by enlarging the pore size on the cell surface so that the DNA molecules can enter. Also, due to the loss of lipids and proteins, the membrane is depolarized, further reducing the repulsion between the DNA molecule and the membrane. Also, the cell density can affect the efficiency of transformation, and it has been reported that maximum competency is observed at cell densities ranging from 107 to 108 cells per ml in the log phase. Okay. Now, uh, there's a question whether the pores through which the foreign DNA enters the recipient cell are formed by the calcium treatment or if they are naturally present. Now, there are already existing natural channels which are called Bayer's bridges in the membrane that can serve as potential pathway for DNA uptake. Okay. either through the pores that are formed. The DNA can enter via those pores already, or via those pores, or there might be already existing channels on the cells, like recipient cells we are talking about, through which the DNA can enter inside. Okay. Now, um, in 1983, Hanahan they stated that competent cells have different sites or channels, and these sites and channels can have an independent chance of taking part in the uptake of DNA, hence participating in the process of transformation, basically taking any kind of foreign DNA material. Now, all the cells, whether they are competent or not, compete for the uptake of plasmid, but if only competent cells are used for the transformation, definitely the efficiency will be increased up to 50 folds, right? So the DNA uptake factor is the sum of all the probabilities of DNA uptake through each channel. The channels that we are talking about either already present or the pores that were created on the cell surface via the treatment of different kinds of by the treatment using different kinds of salts, divalents, calcium or magnesium salts. So it was also reported in a study that the chances of transformation are not increased by increasing the concentration of DNA, but by increase in the number of channels through which the DNA uptake can take place. Okay. Also, calcium has a dual role in this process, in the process of transformation. One is it is neutralizing the charge, definitely, but it is also weakening the cell membrane to produce invaginations, which eventually helps in forming the pores and DNA can be taken inside. Okay. Now, it was known earlier that divalent cations can help neutralize the charge. The complex ions can also serve to produce a static force of attraction within the DNA molecule. Now, this leads to folding of DNA into a compact ball-like structure that facilitates its entry into the cell. The DNA being a large, suppose we were talking about, I think in the last tutorial session, we talked about different forms of plasmid DNA, right? It is circular, double-stranded circular DNA, but it can take up different conformations, right? So it can have a super coiled form, which is like really coiled onto itself. So the overall hydrodynamic radius that we are talking about, that is so small that it can easily pass through the pores. So similarly, if the DNA is folding into a compact ball, it will be more easier for that particular structure to move inside the cells, okay, through the pores or already existing channels. A supercoiled ball-like structure of the plasmid definitely will have more chances of entering the competent cells. However, if the size of the DNA approaches the size of the pore, definitely the probability will decrease. Probability of the particular DNA to get inside the cell will decrease. Now, there are certain chemicals like spermidine or other trivalent salts where the size of the ball-like structure of DNA might exceed the size of the pores in the cell membrane, and this particular problem can be solved by altering the physical parameters like heating and cooling cycles. So if something now some kind of DNA has to be has been treated with some kind of chemical, then eventually the chemical is also attached with the DNA. So overall the DNA, foreign DNA's size or hydrodynamic radius increases. It becomes difficult for the DNA to get inside the recipient cells through the pores. So in that case, what can be done is one can play around or suppose increase the heating and cooling cycles so that eventually the recipient cells have enough time as well as enough enough surrounding conditions for it to form enlarge its pores for the uptake of foreign DNA. Whether using divalents or trivalents, their concentrations need to be optimized such that all the phosphates of the DNA are not rendered inaccessible, because some of the parts of the DNA have to adsorb onto the cell surface, and for that free phosphates are required. Okay. So the general protocol that is usually followed in laboratories that we usually perform as researchers, what we also do is it's a general standardized protocol of competent cells, forming competent cells, and then transformation, which has till date proven to be good enough, okay, and efficient. The transformation efficiency is greatly affected by the type of the host cell, because we talked about bacterial cell, plant cell, fungal cell. These have an additional cell wall as well. So the host cell type can have different cell surface structures, especially in relation to the O polysaccharides that protrude out of the cell surface. Now these polysaccharide surface structures interact with divalent cations and the DNA, thereby making the cell competent for transformation. Now different strains of this bacteria, E. coli, have been reported to show variance in transformation efficiency uh, owing to the chemical properties, different chemical properties on its cellular envelope. Okay, so the cell surface composition also can affect the transformation efficiency. How efficiently can it really take up the DNA? The cell surface composition can also affect it or enhance it, or reduce the efficiency. A very dense O polysaccharide will become a deterrent for the DNA to pass through. It will affect the transformation efficiency in the sense by lowering the efficiency. However, it has also been claimed that extensive removal of lipopolysaccharides by excessive ethanol pretreatment reduces transformation efficiency. So, the authors came up with the suggestion or hypothesis that DNA first attaches to some external component of the cell membrane first, and then the cell membranes, the lipids, like the constituents of the cell membrane, the lipopolysaccharides, proteins, etc. The it's basically its movement and fluidity then helps taking up the DNA inside the cell. The DNA first actually attaches onto some component. Okay, because why did they come to this hypothesis? Because when they completely removed the, like, if the LPS composition is too high for a cell membrane, then the DNA couldn't really pass through. However, if they removed the LPS totally, then also it reduced the transformation efficiency. So that's why they came up with the hypothesis that DNA must first be attaching to some component or constituent of the cell membrane, and then that is eventually helping in the movement of that foreign DNA inside the host cell. Along with the density, the composition of the O polysaccharide also plays a role in the reception of the incoming DNA molecule. Calcium ions also interacts with the membrane, and it has been seen that 100 millimolar calcium chloride concentration, or concentration of calcium salt, almost all calcium is absorbed by the cell's membrane phosphatidylcholine and phosphatidylserine. Okay, therefore the membrane properties can play a major role in DNA adsorption. Okay, so the evidences clearly indicate that the physical and chemical treatments basically referring to the divalent salts, that we are magnesium and calcium salts, and physical is mostly the temperature variations that are being used. During transformation, that is the temperature imbalances and calcium chloride treatment help deal with the barrier to DNA uptake, such as charge repulsions and pore sizes. Okay, so this is what has been shown here diagrammatically. Don't know if it's visible properly. So here what is happening is, this is the foreign DNA, this is the host cell we are talking about. In this example, it's bacterial cell. Now it has been shown, this particular cell membrane has been enlarged. Now what is exactly happening upon treating it with calcium salts, as well as giving it temperature, treating it with different kinds of temperature? So the DNA is negatively charged membrane is there. So negatively charged DNA, negatively charged membrane, calcium salts are neutralizing the charges on both these parts here. Okay, and similarly, when temperature treatment is given, there's lipid loss, right, at higher temperatures, eventually creating pores, and protein loss at lower temperatures. Eventually, this creates pores in the cell membrane, and the DNA, by this action, the DNA can enter inside the cell membrane. The DNA gets transferred inside the cell. Okay, this is just a pictorial representation. Now, how do you really prepare a competent cell? Bacterial cell, the competent cells in this case, is incubated with divalent cation, calcium chloride, manganese chloride, or rubidium chloride for 30 minutes at 4 degree. And during this process, the cell wall of treated bacteria swells up and gathers factors required for intake of DNA docked on the plasma membrane. Actually, competent cells are really fragile. Okay, and so what we have to be very careful is during preparation, while following the protocol, the centrifugation speed shouldn't be too high. Okay, now when you centrifuge it, there will be cell pellet. Okay, now the cells, when these competent cells are pelleted down, they have become too fragile. So usually, most of you might have used a pipette, or even if you haven't yet, what is done is like there's something called a pipette from through which the tips are like conical micro tips are there. Okay, now it has pointed tips through which you take like as low as you can take up as low as microliters of samples, liquid samples through the pipette. Okay, now while taking a competent cells, we have to first the cell competent cells are pelleted down by centrifugation, and then it is resuspended in these solutions. Okay, so these solutions are like while resuspending the cell pellet, what happens is the tip is micro tip is too conical and sharp. So what it can also do is it can affect the competent cells. It can shear the competent cells. The cells are too fragile after the salt treatment and temperature treatment. Okay. So what happens here is we have to be very careful. Usually people use the cut tip, broadened. So if you simply cut it with scissors, what happens is the tip, the sharp tip, conical tip widens. Okay, and then you eventually take in the cells and resuspend the cells via that broadened tip, and then the whole thing is to be really careful while handling the cells, like very gentle. We have to be very gentle with the cells because they become too fragile. And then this is stored at -80 uh, in glycerol, because glycerol is a cryoprotectant. Okay, so it won't really harm the cells. Like when you have to use it, this is completely frozen at -80, so it will eventually thaw. The cells will thaw and form liquid, finally liquid suspension. So that liquid suspension, while forming the liquid suspension, if when it's freezing, the ice crystals can eventually harm the cells. So that's why glycerol, this cryoprotectant, is added so as to keep the cells intact, competent cells intact. Okay, so and then this is the transformation protocol. Now, ampicillin sensitive E. coli cells in log phase of growth are transferred to cold calcium chloride solution. Then, ampicillin resistant plasmids are added to experimental cells only. Uh, Basically, these are the competent cells. Okay, this is what is being talked about, competent cells, and these competent cells do not harbor any kind of plasmid that is giving them antibiotic resistance. Now, the foreign DNA, these competent cells have to take up a foreign DNA. Okay, which is foreign plasmid or a DNA. That plasmid actually harbors ampicillin. Ampicillin is an antibiotic. This antibiotic resistant gene. Okay, now, these plasmids that are harboring the ampicillin resistant gene has to be put inside these competent cells. Now, if the cells which eventually take up the ampicillin resistant plasmid can grow in ampicillin containing medium, but the cells which have not been able to, which aren't really competent enough to take up these ampicillin resistant plasmids, those won't be able to grow in ampicillin containing medium. Okay, so the cells are already there on in ice. You have the plasmid DNA as well, foreign DNA. This is added and then incubated on ice and then given a heat shock at 42 mostly. That's what I was talking about earlier. And then this is given, the cells are again then incubated and then plated on agar plates containing ampicillin. Okay, ampicillin here is acting as a selectable marker. Okay, so as to differentiate between the cells which have taken up the plasmid or recombinant DNA and those which could not take up the foreign DNA. Now, how do we really determine the transformation efficiency? Uh, so whenever you are plating it on an agar plate, you will be able to see bacterial colonies. Okay, so the number of colonies that are there and then divided by the microgram of DNA that you have added in the very first step to the competent cells into a final volume at recovery divided by the volume plated. Now, after giving it heat shock, so the cells are now only the DNA. At this point, only the DNA has gone inside the cells. Now, it's still a small volume of competent cells that's there. Now, the cells that have taken up the DNA has to reproduce, has to produce, like those cells have to produce their progenies so that we can harbor many more or culture many more cells with having the plasmid of our interest, having the gene of our interest, right? So for that, after this step, it's not shown here. After this step, what is done is simply add the liquid media, small amount, 1 ml or 900 microliters of media into this particular suspension, and then you let the cells multiply, because now the small amount of cells have a good amount of food and nutrition source for them to multiply. And then after it multiplies, so this particular uh, addition of LB media or culture media to the mixture is called recovery media. Okay, now it is then incubated and we let the cells grow, and then from that recovery medium some volume is plated on the agar plate. So that's what here it's mentioned. What is the final volume at recovery? Suppose 1 ml, and then how much did you take out of the final recovery volume and plated on the agar plate? That will eventually give you the transformation efficiency, denoted by the number of transformants, basically the number of cells per microgram. Okay, number of colonies, bacterial colonies per microgram of plasmid DNA. Okay, now the amount of the plasmid that is used in the transformation is in micrograms, and the amount of plasmid that can be used for transformation is 10 nanograms. 50 nanograms can also go depending on what type of host cells are we talking about. Okay, what is transformation efficiency is also affected by the final volume of the cell suspension at recovery. That's what I was talking about after the heat shock. This includes the volume of the competent cells, already the mixture of competent cells as well as the DNA. And the competent cells have taken up the DNA, and it already, suppose usually the volume of competent cells are like either 50 microliters or 100 microliters, and the volume of cells plated on the nutrient agar, definitely how much out of the whole 1 ml media or 500 microliters or 900 microliters media, how much are you taking and then plating on the agar plate? That will eventually uh, like that has to be taken into consideration, otherwise you won't be able to calculate the total number of, like, the absolute number of cells in there. Okay, since most of the time we will not plate the entire volume, it is not really possible to plate um, like 1 ml of culture. Usually we take 50 microliters or 100 microliters and then plate it on an agar plate. Okay, and definitely the number of colonies that eventually grow on the agar medium, that also determines how much is the transformation efficiency. Okay, so this is an example. Suppose you transformed 1 microliter of this much plasmid. That means if you are transforming 1 microliter, this is the concentration of the plasmid. So if you are adding 1 microliter, that means you are adding 0.01 nanogram. Okay, into 100 microliter of competent cells. Now you added 900 microliter LB medium. This is the recovery medium that we were talking about earlier. 900 microliter to 100 microliter of competent cells to get a total reaction volume of 1 ml. And then after letting the cells grow, we plated 100 microliters of the transformed cells. And finally, we get 450 colonies. So what is the transformation efficiency? Now, so what is the nanogram of how much is the nanogram or in general the amount of DNA plated? 1 microliter into 0.01 nanogram into the concentration of the plasmid. Okay. Now, how much has been plated? 100 microliters, right? 100 microliter plated, and total reaction volume was 1000 microliters, right? So total DNA plated is 0.001 nanograms, right? If it's 0.001 nanograms, so what will be the transformation efficiency? Because finally we had 450 colonies on the agar plate, so the transformation efficiency will simply be 450 colonies into the nanogram amount of DNA. Now if we have to, this is a nanogram, because we have to show it in like micrograms in quantities, number of colonies or number of transformants per microgram of DNA, then the nanogram has to be converted into microgram unit. Okay, so this is how you will be calculating the transformation efficiency. So now what are the methods of transformation? One is like methods of introducing foreign DNA into the host cell. One is electroporation. So this is the host cell, um, this is the cell membrane, and then this is the DNA or nanoparticle or any kind of foreign particle that has to be introduced inside. So material is introduced to the fluid around the cell first, and then pulsed power generates an electric field that opens up the pores in the host cell membrane, and the material, any kind of material, foreign DNA, nanoparticle, etc., can be introduced inside the host cell. And when the pulse is removed, the electric field is stopped, like there's no electric field as such, so the pores eventually close on the membrane, pores on the membrane close, and the material remains inside. Okay, this is the using electric field we are introducing the foreign material inside the host cell. This is electroporation. Factors that affect transformation efficiency: plasmid size. All right, forms of DNA, we were looking at ball-like structure of DNA, supercoiled versus relaxed DNA. So definitely the supercoiled one will have more chances of getting through the pores on the cell surface. Definitely the growth of the cells, competent cells, if the competent cells are good enough, they are really competent enough in a very literal sense, then they will be able to take up more amounts of foreign DNA. Transformation protocol, if we are following it properly, the heat shock time, the temperature, the growth media, the chemicals being given, etc., if there are any contamination or not, and also if the DNA is intact or pure enough that we are introducing inside, that can also affect the transformation efficiency. Okay, another is chemical transfection techniques. It what happens is the foreign DNA of our interest is coated with a chemical. Okay, mostly a polymeric compound, or the whole complex in question is coated with a treated with chemicals or a polymeric compound for it to um, precipitate, and then this precipitate actually interacts with the cell membrane of the host cell, and then eventually this complex is taken out. Now there are multiple chemical compounds which actually help the DNA uptake into the mammalian cells. Okay, this is one of the chemical transfection techniques for taking up DNA inside, for introducing DNA, foreign DNA inside mammalian cells. Okay. One is calcium phosphate method. What happens is DNA is mixed with calcium chloride in a phosphate buffer and incubated for a certain time. And then the mixture, which is called the transfection mixture, is added Um, and then the DNA calcium phosphate complex that we looked at earlier, chemical transfection method. This is one of the examples where the DNA calcium phosphate complex is formed. It forms a precipitate and then deposit on the cells as a uniform layer. And this particular precipitate is then taken up by endocytosis into the internal storage of the cell. Then eventually once inside the cell, the DNA escapes from the precipitate and reaches the nucleus through an unknown mechanism. Okay, and this method is suited to the cell growing in a monolayer or in a suspension, but not for cells growing in clumps. Because if the cells are clumped already, then the calcium phosphate complex that has been formed along with the DNA, that won't be able to cover uniformly the cells, right? If the cells are clumped already. So that's why if the suspension is homogeneous kind of, or if it's a homolayer of cells, then this DNA calcium phosphate complex method of transfection is an efficient one. Okay. The disadvantage of calcium phosphate method is severe physical damage to the cellular integrity. Why? Because the cells are actually coated with this precipitate. Okay, this particulate matter that settles on the cell surface, which also can lead to reduced cellular viability as well as cytotoxicity to the cells. It can eventually kill the cells as well because it's a thick precipitate or particulate. Sorry, am I able to, are you guys able to hear me? I lost connection just now. Hello? So there's a load shedding here. So if you guys are able to hear me, can you guys just unmute yourselves and let me know that? Okay, I will continue teaching. Now there's a polyplex method, which is an alternate method for DNA, where DNA is complexed with a chemical agent to form a soluble precipitate or polyplex through electrostatic interaction with the DNA. Okay, so there can be different kinds of complexes. Again, it's basically a DNA complex forming a complex with DNA, and then the soluble aggregates of DNA with polycationic complex is readily taken up by the recipient or host cell, and it reaches the nucleus for expression. Okay. And then there's liposome and lipoplex method. This is another approach of DNA transfection in animal cell to pack the DNA in a lipid vesicle or liposome. In this approach, DNA containing vesicle will be fused with the cell membrane and deliver the DNA to the target cell. Preparation of liposome and encapsulating DNA is a crucial step to achieve good transfection efficiency. Okay, I think the power is back now. Hopefully, I'm still connected. Yeah. Okay. Now there's transduction as well, where mostly phage uh, is the bacteriophages are used. The viral mode of infection is used as a medium to transfer a foreign DNA. Okay, so what happens here is phage injects DNA, then the phage enzyme breaks down the host DNA, and then the cell creates new phages, including the phage and the host DNA. And the transducing phage can insert the donor DNA as well. And here, the donor DNA is included in the recipient cell's chromosome due to recombination. Okay, this is another way which is called the transduction. Okay, and screening of recombinant clones, we have been talking about selectable marker. So one can be the enzyme we looked at in the previous tutorial session, beta galactosidase enzyme. Then there's antibiotic resistance gene, we have been talking about it, or some kind of phenotypic changes that can also be taken into account for screening the cells, only those cells which have taken up the foreign DNA or the recombinant DNA. So one is definitely antibiotic sensitivity. Now, suppose this is a particular recipient like host gene and recipient cells gene, and this is the DNA or gene of our interest. Okay, and this particular recombinant plasmid has ampicillin resistance. It shows ampicillin resistance. So in an ampicillin containing medium, this particular um, plasmid can grow inside if the cells have been able to take up this particular plasmid. Okay, but if this particular gene hasn't been inserted, okay, then what can happen is uh, this also can get inside the host cells, but it won't really be growing in an ampicillin containing medium. Okay, so antibiotic resistance genes and their mode of mechanism. So ampicillin, kanamycin, tetracycline, chloramphenicol, there are many other um, antibiotics as well. So out of these four are very commonly used. And ampicillin actually produces, the cells can produce beta lactamase, which leads to degradation of ampicillin. That's how the cells become resistant to ampicillin antibiotic. Okay. For kanamycin, there's neomycin phosphotransferase 2. The kanamycin resistant gene produces this particular product, which modifies the kanamycin covalently, and the cells become resistant to kanamycin. Then there's tetracycline. Usually the tetracycline resistant gene produces some kind of ribosomal protection proteins, and what actually happens then is the tetracycline antibiotic gets outside, is actually thrown outside the bacterial cells, the ones which have been able to take up the tetracycline resistant gene. Then there's chloramphenicol. Um, the chloramphenicol resistant gene encodes for chloramphenicol acetyl transferase, and which eventually modifies the chloramphenicol to acetyl chloramphenicol and isn't able to uh, really show its antibiotic mechanism. Can't have can't have its action on the resistant cells. Okay. Insertion and inactivation, we looked at it in the previous tutorial session in great detail. I will go through it briefly. So what happens is a foreign DNA is cloned within the coding gene responsible for a particular phenotype. Suppose we talked about in the previous tutorial session about the antibiotic resistant phenotype. Okay. As a result of insertion, now if the insert DNA is actually in the inside the gene encoding for antibiotic resistance, right? So if the insertion is in that particular region, then the cells will lose its antibiotic resistance. If the insertion hasn't been in that particular region, then the cells will have an intact antibiotic resistance mechanism. Okay, so this is one of the examples. So which is called insertional inactivation. Uh, and so suppose this is the very first plasmid we looked at, recombinant plasmid, having both tetracycline as well as ampicillin resistance genes, right? PBR322. Now, if a gene fragment is cloned in this particular region, okay, uh, like if so, this harbors this PBR322 recombinant plasmid harbors these two antibiotic resistant genes, ampicillin and tetracycline. Okay. Now, if a gene fragment will be cloned, it will disrupt in this particular region, then it will disrupt the ampicillin resistant gene. Okay. And then what will happen is the particular cells with the insertion here in this particular region will eventually die if the culture medium or if the cells having this particular plasmid is exposed to ampicillin, because eventually the ampicillin resistance gene is disrupted, right? But the original plasmid, if the some cells have been able to take up this original plasmid, then it can easily, very happily grow in an ampicillin containing medium, right? Because it has a fully intact and functional ampicillin resistant gene. So, but if a particular gene is inserted in this particular region, it will disrupt the functionality of ampicillin resistant gene. But tetracycline resistant gene is very happy. It is completely functional. Like we don't, we haven't yet targeted this particular gene, so it's fully functional. We just disrupted the functionality of the ampicillin resistant gene. Okay, so there are two antibiotics here at play. Now, what is happening is the clones will be ampicillin sensitive, but um, but it will be tetracycline resistant. Now, to select the clone, first the transformed cells are plated on a tetracycline containing medium. So any type of cell which has taken up this plasmid can grow on a tetracycline medium, whether it's ampicillin resistant or it's ampicillin sensitive. It has a functional tetracycline resistant gene. So all the cells which have taken up this plasmid can grow on a tetracycline containing medium. Now, out of the tetracycline containing medium, the ones that are growing on ampicillin containing medium are the negative clones, because the gene that we targeted here, that we actually wanted to insert here, didn't really get inserted, and that's why there's a functional ampicillin resistant gene. If the gene would have been inserted here, suppose in this particular area, then the functionality would have been disrupted for this particular ampicillin resistant gene, and it would have been um, killed the ampicillin like killed the cells which are which have the insert, which harbor the insert here. Okay, so this is what is called insertional inactivation. You are basically inactivating or disrupting the gene here, antibiotic resistant gene here. Okay, this is the same thing that we talked about earlier. uh, so the ones that are growing on tetracycline containing medium are the transformants, and the ones growing on the ampicillin, are not growing on the ampicillin containing medium, are the recombinant ones. Okay. The same way we have the alpha complementation process, where lactose in the surrounding environment triggers the laxioperon in E. coli, and which leads to the production of beta galactosidase, and this enzyme metabolizes lactose. I think it is the same slides from the previous tutorial session. So where this particular mutation, M15 mutation, delta M15, produces omega peptide without the N terminal part, and there's the N terminal part is called alpha peptide. Now, individually alpha peptide or omega peptide can't be functional, can't form a functional beta galactosidase, but whenever the alpha peptide and the omega peptide both are present, this will actually form the functional beta galactosidase enzyme. Okay. So when these two peptides are expressed together, functional beta galactosidase is formed. So this is what is actually used as a marker for recombination. What happens here is multiple cloning site is present within the LAGZ sequence. The sequence can be nicked by restriction enzymes to insert the foreign DNA, and when a plasmid vector containing foreign DNA is taken up by the host E. coli, alpha complementation does not occur because the plasmid DNA containing foreign DNA, plasmid vector has the foreign DNA insert, so it has insertional inactivation. So functional beta galactosidase enzyme is not produced. If the foreign DNA is not inserted, or if it inserted at some other location other than the site where we actually wanted it to be inserted, the LAGZ gene in the plasmid vector complements the LAGZ deletion mutation in the host E. coli, thereby mainly the LAGZ gene in the plasmid vector complements the LAGZ deletion mutation in the host E. coli, where both alpha peptide and omega peptides are formed, thereby producing a functional beta galactosidase enzyme. Okay. We again looked at blue-white screening, where this chromogenic substrate X-gal is used and beta galactosidase acts on it to produce one insoluble blue pigment, a precipitate, which can be helped to which can be used to differentiate between recombinant and non-recombinant clones. Okay, this is how it looks on a plate. Okay, so the blue colonies are the ones which do not have the insert of which do not have the recombinant DNA, but the white colonies are the ones which have been able to take up the DNA. Okay, so this is the one that we were talking about. This is the foreign DNA. This is the plasmid vector. Now, competent E. coli cells have harbor this particular deletion where the N-terminal part is not encoded, only omega peptide is encoded. And this particular gene, this insert, this particular plasmid has the particular gene encoding sequence which encodes for the N-terminal part of beta galactosidase enzyme, which is the alpha peptide. Okay. Now, if this is there, just already in this is there, which already this only plasmid vector encodes for alpha peptide. The cells, competent cells, have this delta M15 construct, which can only encode for omega peptide. And when alpha and when these cells are put inside these competent cells, then alpha and omega complementation occurs, and then it is producing a functional beta galactosidase enzyme. Now, this is the general scenario. Now, you want to insert a foreign DNA. Okay. Now, if the foreign DNA is inserted within the LAGZ gene in the plasmid vector, what will happen? It is disrupting the functionality of the LAGZ gene. So, functional alpha peptide won't be produced, right? So, if functional alpha peptide won't be produced, competent E. coli cells which have this delta M15 deletion will be producing omega peptide. Definitely, that is already there. But unless the alpha peptide is produced, it won't be able to complement and form a functional beta galactosidase. So, if the foreign DNA of our interest is inserted in this particular region, then there will be no functional alpha peptide, hence no functional beta galactosidase. So, the chromogenic product X-gal, chromogenic substrate X-gal won't be producing the blue colored precipitate, hence the colonies will be white. If the insert is somehow the foreign DNA has been inserted, has been recombined in the plasmid vector, but at a different region altogether, then also the LAGZ gene is actually intact here. So it will produce a functional alpha peptide which will eventually complement the omega peptide, thereby producing a functional beta galactosidase which will act on this chromogenic substrate X-gal to produce blue colored precipitate. Okay. And if there's the plasmid vector hasn't inserted any foreign DNA like anywhere inside it, then also definitely the LAGZ gene encoding for the alpha peptide is intact, and it will eventually produce blue precipitate. Okay, so this is a visual screening process here. Now, complementation of mutation, one is positive selection and one is negative selection. So, in positive selection, the host strain isn't growing on the media lacking the functional gene, but if the host is transformed with the recombinant DNA with the foreign DNA, then the host cells can actually eventually can grow in the given media. Okay, because the if the host cells have been able to take up the recombinant gene, then that particular gene actually can supply the product which the cells require to grow in a certain type of medium. Okay, growth medium. Then there's negative selection as well, where a chemical compound is added to the medium, growth medium, which is converted to a cytotoxic agent in the presence of the gene product, like the gene product which is encoded by the recombinant DNA, and as a result, it doesn't allow the growth of wild type cells. But the host strain transformed with the recombinant clone has non functional gene product and grows in the presence of the compound in the growth media. Okay, now suppose this is a particular gene, URA3, and then it encodes for this particular decarboxylase enzyme, and this converts fluoroorotic acid to a toxic fluorodeoxyuridine, and if this particular toxic product is formed, then there are no cells growing, no colonies. But if there is a foreign DNA, foreign gene of our interest in this particular site, then what happens is this particular enzyme, a non-functional decarboxylase enzyme is there. So what happens is eventually it won't be producing this particular toxin or toxic product, and hence we can see the growth of colonies here, right? So this is how this is one type of selection that is taking place. Now there's reporter gene assay as well. This is the native, like this is a natural scenario where there's a promoter, there's the natural gene, mRNA is transcribed, which further encodes for the protein. This is the endogenous gene product. Now what happens in a recombinant cell, you have a promoter region definitely, and then a reporter gene, like which encodes for certain kind of chromogenic or fluorogenic product. Okay, and then you have some kind of enzyme like beta galactosidase or some kind of fluorescent protein like green fluorescent protein or yellow fluorescent protein, etc., or some kind of other enzyme like luciferase which acts on certain substrates giving fluorescence, okay, emitting light, right? So this is one of the assays which is called luciferase assay, wherein you have luciferin substrate, okay, and then luciferin substrate in the presence of like luciferin substrate in the presence of ATP forms this luciferyl adenylate adduct, and then it in the presence of luciferase enzyme, CO2 and AMP is released and it gives off light, thereby forming oxyluciferin. Okay, hence, and this particular light this particular light can be detected via luminescence, and then reading can be taken. And then now we have the cells with the recombinant DNA. The host cells have taken up the DNA. Now, how do we really know that, okay, our gene of interest is actually inserted in the plasmid vector, right? So, for that, we do something called sequencing or DNA sequencing. The very traditional way of doing sequencing is dideoxy chain termination or Sanger sequencing. So this was actually developed way back in 1977 by Frederick Sanger, where what is being done is usually it's ATGC, right? Deoxynucleotides are added one after the other to form a nucleotide chain, right? But in this scenario, in Sanger sequencing, dideoxynucleotides or DDNTPs, these are actually used where usually for elongation, for any kind of polymerization, right? For DNA polymerization, you need 3' hydroxyl, free 3' hydroxyl group. But in case of dideoxynucleotide triphosphates or DDNTPs, this 3' hydroxyl group is absent. So that's where the chain polymerization terminates. The chain can't extend if any kind of, at any point, DDNTP is incorporated, right? So what happens is, in a typical sequencing reaction, 4 different DDNTPs are taken into 4 separate reaction tubes and then analyzed on a polyacrylamide gel electrophoresis. Okay. So if the DNA polymerase utilizes DDNTPs as nucleotide, suppose the um, template has A there, so complementary nucleotide will be T, but then uh, what happens is, if DDTTP is incorporated, then after the T, there won't be any other addition of nucleotide because of the absence of 3' hydroxyl group. All right. So that is where the chain terminates. Similarly, for other type of sequences, once the DDNTP is incorporated, then the chain termination will take place, and then based on the different kinds of different lengths of the fragments, the complete sequence of the DNA can be deduced. So this is, suppose, the complete sequence. We don't know what is the sequence here, but so what happens is, you have 4 reaction tubes: A reaction, T reaction, G reaction, and C reaction. Basically, A reaction is having any kind of whatever is ending with A. Similarly, T, G, and C reaction. So this is how the gel looks like. Okay. If the fragment is stopping at this point, then it has A here at the end. Okay. And the complement, like if it is stopping at T, then the complementary is A. So this will be the area, like A nucleotide in that sequence. Similarly, it goes on. Okay. Then there's Maxim Gilbert method, which is also used on based on radio labeled nucleotides. Okay. So what is happening here is, it was also developed in 1977, where the chemically modified nucleotides, 3' or 5' radio labeled DNA is treated with base specific chemicals, which then randomly cleave the DNA at their specific target nucleotide, and then these fragments are analyzed on polyacrylamide gel, and an autoradiogram is developed. And then the fragment with terminal radio label appears as a band in the gel. So this is what happens: G reaction, G plus A reaction, T plus C reaction, and C reaction. Now, this is the gel and autoradiogram that eventually comes up. What is happening here is, the fragment in the G lane here is read as G, whereas the fragment present in G plus A, but which is not there in G, will be read as A, right? Similarly, if something is present in the C lane as well as in the C and T lane, or then it is C, but if something is present both in C lane but not in is present in C plus T lane but not in C lane, this one will be read as T. Okay. To get the DNA sequence, the band with the lowest molecular weight is read first, followed by the next band in the 4 lane. Okay. So here it is, this band is present in both G as well as G plus A reaction. So it is G. Okay, this is the lowest band. The next band is here. Now this is present, this band is absent in G reaction, but this band is present in G plus A reaction. That means this is an A. So after G, it comes GA. Next comes this particular band, right? Now this particular band is present in which tube? T plus C reaction tube. But this band is absent in C reaction tube. So that means this is ending at thymine. Okay, thymine residue. So GAT. Now the next molecular weight is this one. This fourth band is this one. Now both the lanes, like both the reaction tubes, T plus C as well as only C, are having this particular band. So this is a C residue, right? So this is how eventually we build up the complete uh, nucleotide chain or nucleotide sequence. Okay. So let's look at few questions. quickly. So which of the following agents is used to make E. coli competent cells? We looked at it very like we looked at quite in detail, which is option C, calcium chloride. While making competent cells, it is essential that the bacteria is in which particular phase? It is option B, log phase. Transformation efficiency in E. coli is highest for even whatever is more compact, that will be more efficient in transforming. So the option will be option A, supercoiled DNA. Which of the following components block the non-specific sites on the cell wall and increase the efficiency of plasmid uptake in yeast cells? Okay, so for yeast cells, it is lithium acetate, or mainly this is their either lithium acetate, PEG 3500, single stranded carrier DNA, and foreign plasmid at 42 degrees Celsius for 40 minutes. So this carrier DNA is actually added to let it form like let it form a complex which blocks the non-specific sites on the cell wall, and the plasmid, the ones which we want to introduce inside the cell, is ready for uptake. Okay, so the answer will be option B, single stranded DNA. Okay, the single stranded carrier DNA actually blocks non-specific sites on the cell wall. Okay, which of the following enzyme could be used to partially digest the cell wall of yeast cells? Answer is option C, zymolase. In the calcium phosphate method, where we talked about the DNA and the chemical agent forming a complex, the mammalian cells uptake the DNA calcium phosphate complexes by the process of what happens eventually is endocytosis. Option A. We looked at it in the chemical transfection method. Okay. The method in which viruses are used for transformation in mammalian cells is option C, transduction. In the bactofaction method, dash is commonly used for transformation of plant cells. So in bactofaction, what happens is the plant Agrobacterium tumefaciens is used. So this bacteria is taken up by the host cell through phagocytosis and entrapped in a membranous vesicle known as phagosome. Okay. Then the bacteria can escape from the phagosome and get lysed to release the DNA eventually in the cytosol. Okay. So eventually the plant cells can have the recombinant DNA. So the option is option B, Agrobacterium tumefaciens. In insertional inactivation mechanism, the successful cloning of the gene of interest in the LAG-Z gene will result in dash colonies. So it will be colorless or white colonies, because something that is inserted in the LAG-Z gene will render it non-functional, right? It won't be able to encode for the alpha peptide. So the option will be option D. So if the gene of interest is inserted anywhere outside the LAG-Z gene, or it hasn't been inserted at all, then the LAG-Z gene will be functional. It will encode for a functional alpha peptide. Alpha and omega will combine together to form a functional beta galactosidase, which eventually will act on X-gal chromogenic substrate to form blue colored precipitate or blue colonies, right? But now they are talking about, the question talks about successful insertion of the gene of interest in the LAG-Z region. So that will eventually lead to white colonies. Okay. So option D. The cytotoxic compound added to the selection media for URA3 mediated negative selection is, we talked about it, and the cytotoxic compound is, this is the compound formed, this is the product formed, and the enzyme actually acts on 5-fluoroorotic acid. Okay, so option will be option A. This is the product that is formed. Dideoxynucleotides are used in which type of sequencing? We just looked at it. It is option C, Sanger sequencing. So in the G reaction of Sanger sequencing, the DNTPs will be, so there will be one dideoxynucleotide and others will be normal deoxynucleotides. Okay, but when we are talking about the G reaction of Sanger sequencing, then the DNTPs will be this one. All will be normal ones except DDGTP. Okay. So if there's DDATP, then that will be an A reaction. Okay. Which of the following reaction is catalyzed by the gene product of 4A gene? So this is from the lecture slides only. So 4A gene um, has the encodes for the gene product alkaline phosphatase, and it catalyzes, like leads to the release of inorganic phosphate. Okay, so option C will be the correct answer. Now, in general, this is about the viral cycle, viral lytic cycle. So, during an infection cycle, virus undergoes a lytic and a lysogenic cycles. Okay, during the lytic phase, this is responsible for the lysis of the host to release the viral particle, whereas lysogenic stage allows the replication of virus without the lysis of the host. Right. So there is the CI gene which encodes for CI repressor and which is then responsible for the formation of lysogens, right? Now, in the presence of a functional CI, what happens is the plaques contain unlysed host cells and has a turbid appearance, whereas when the CI repressor is not there, then there's no plaque and there's no turbid appearance. This feature can be used to screen the clone to detect the functional CI, which is the absence of any clone, or the absence of CI, which suggests the presence of the recombinant DNA along with the region of our interest. So this is what the question is. The CI repressor, when it is active, actually represses the lytic phase. Now this is something related to the transformation efficiency. We looked at it in detail. Now during a transformation, this much amount of DNA, microgram of DNA was used to transform this much volume of competent cells, right? And the cells were recovered by adding 900 microliter of LB media, and the final volume is 1000 microliters, which is 1 ml of total reaction volume. Only 50 microliter was plated, and eventually 500 colonies were obtained. So the efficiency will be how much microgram, how many transformants, 500 transformants into how much microgram of DNA was taken up. Okay, so it will be 0.001 into what is the final volume, 1000 microliters, by the volume that was plated by 50. Okay, so the option will be 10 to the power 7 colony forming units or CFU per microgram of DNA. Okay, this will be the transformation efficiency. So this is it for today's session. Thank you for joining. There was an interruption in between. Sorry for that. I lost connection. Kriti Sinha wants to join. Actually, it's the class is over now. So whenever you guys, anyone is wanting to join the meeting, the people who are already there, you guys can unmute yourselves and let me know, because I won't be able to know if someone is waiting for waiting to join the meeting. Okay. Thank you all. I will upload the recording of this session completely in YouTube, and I will share the lecture slides as well. Okay. Thank you for joining, everyone.